Hi, in this video I'm going to demonstrate two ways of hemolyzing red blood cells. In part 1 we are going to do an osmotic fragility test, and in part 2 we will use a plant steroid called digitonin. In order to perform an osmotic fragility test, we will set up a concentration gradient of sodium chloride starting from the physiological salt concentration of 145 millimolar. So I'm going to prepare 6 tubes, and then I will add decreasing amounts of sodium chloride and top it off to a final volume of 1 milliliter with distilled water, before adding 5 drops of blood to it. Now, why are we doing this? And what are we trying to measure in the first place? We know that red blood cells are stable at 145 millimolar sodium chloride because that's the level that they are found in the blood. If we decrease the salt concentration in the liquid that the red blood cells are in, they will try to compensate by taking up water. Eventually, cells will absorb so much water that they burst and release hemoglobin into the surrounding liquid, which we will be able to see after centrifugation. At a physiological level, the cells should be stable and form a pellet with a clear liquid on top. The more the salt concentration decreases, the more cells will lyse, and we should be able to see a gradient of red-colored liquid. This is called an osmotic fragility test, where we measure the ability of red blood cells to take up liquid without bursting. This can be used to help diagnose certain types of anemias like erythrocytosis and thalassemia. In these diseases, the red blood cells are malformed and can become more or less sensitive to lower concentrations of salt, because the shape of the red blood cell is the primary factor affecting osmotic fragility. So by comparing a patient's blood to a healthy control, you can see if their red blood cells are shaped normally or not. Alright, now that the blood and liquid are well mixed, we can hop over to the centrifuge and spin the tubes for 2 minutes. Remember to always make sure that your centrifuge is balanced. Most centrifuges have markers like the white stripes here to help you find the right position. Okay, so here are our results. We have a decreasing salt concentration from left to right, starting at 145 millimolar, and we can see that we have a nice gradient. Most cells can handle a little pressure, but once the concentration is below 50% of normal, most cells burst even after just 2 minutes. For the second part, we will do another concentration gradient, but this time with digitonin, a steroid from the digitalis plant. It's a detergent and therefore solubilizes lipids in water. It is used in biochemistry to solubilize membrane-bound proteins, precipitate cholesterol, or permeabilize membranes. Now let's have a look at our results for digitonin. Remember, we added increasing amounts of digitonin from left to right, and we do see a gradient again, as expected, maybe not as nice as for the sodium chloride, but still clearly visible.